Okay. So today we're going to be starting on a whole new section for the class. So instead of focusing on the different types of organisms, we're going to start talking primarily about mammals, primarily about humans. So we're going to be talking about the different systems. Before we do that, I want to back up to the chapter on animal organization and talk about homeostasis, because obviously that applies broadly to all of the different systems that we're going to be talking about. So it could be like the circulatory system regulating heart rate, but it could also be the digestive system regulating blood glucose levels. So all of the systems regulate a relatively constant internal environment, and that is what is referred to as homeostasis. So if you look at this word, homeo means same, and stasis means state. So this is technically the same state. Okay. However, we've kind of re-described it because our old ideas about homeostasis are not quite correct from what we now know. And so we now call it a dynamic equilibrium. So does anybody want to try to explain what would be the difference between those two?
If I were to guess, I would say the dynamic part of it would be like kind of sort of on the spectrum of.
There's a range,
right?
And it changes, right? So when we think about like our heart rate, if we think about it, and if I need to walk up the stairs, I don't want my heart rate staying the same, right? I want it to go up so that I get oxygen to my muscles and allow me to do the work. So the dynamic equilibrium is that it varies depending upon the environment, but generally there is a range in which we want to maintain, right? And so that's the idea of homeostasis, the new idea. We still use homeostasis as the word, but we have kind of redefined it as this idea of a dynamic equilibrium. So I'm going to show you a short video that talks about the significance of this as it pertains to heart rate. And the heart rate that they are measuring is over a period of time, and a relatively long period of time. And they're just looking at the variation in the heart rate, even when you're not doing anything active. So it would just be in rest. So, and I just had sound. Happened.
The sound on the video itself is off. Like it's like crossed off at the bottom there.
On the video itself. Oh, there we go. Yeah.
With some. A blue square is a resting end. Touched by an active white square, it lights up and sets to work.
Okay, just ignore this part.
The more action. You can see the graph of activity across the grid. When there aren't many ants, the place is chaotic. But as you add more ants, the activity increases.
Let's see if I can fast forward it.
A distinct rhythm appears. Something new appears. Something new appears. Thing alive. Even my heart, Brian told me, finds its own shifting harmony.
Can everybody hear that okay?
Brian Goodwin sent me to see Ari Goldberger, a cardiologist at Harvard, with some very surprising ideas about health.
It appears that health represents a remarkable balance between excessive order on the one hand, things being overstructured, and complete randomness on the other, where there's, in essence, there will be physiologic anarchy. The healthy systems like to be there. They don't sit still. They're always kind of fidgeting. They're always ready for everything. When people listen to a healthy heart, what they hear is something that sounds quite regular. It's the lub-dub, or they are looking at a cardiac monitor that's showing something that seems to be ticking off impulses, much like a clock. But in fact, when you look at the way the heartbeat is actually changing in a very subtle way, what you see is this extraordinarily complex variability. It was, to us, an amazing surprise to see that the resting heartbeat was, in fact, as complicated as anything that we were able to find elsewhere in nature.
If you measure the beat over many hours, you discover that the heart's rhythm is constantly changing.
Well, I'd like to show you a little game. It's a physiologic game that we developed in the laboratory. And what we're going to see is the pattern that your heartbeat makes if you're just basically sitting around.
This is just a resting heart?
Yeah, is that a surprise to you?
It is.
Yeah, this tracing here is very different from what you might suppose if you
completely
came from the tradition of homeostasis. This should be, if you're resting and you're quiet, this should be a straight line or something approaching that. So one of the ways of trying to convey what healthy physiology is all about is to take this sequence and to translate it, to transpose it, or to map it into a sequence of notes. And so the question is, what does your heart sound like? What is the music of the healthy heart?
That was wonderful. That's a normal heart.
That's a normal heart. And what we're going to see here now is a very different pattern. This is the heartbeat tracing from the patient with heart failure. And what you see here is actually quite different from the healthy. Now, things actually look more organized. They look, in a sense, more periodic. And so let's play it out and see what this sounds like. So which would you prefer to listen to, the healthy heart or this?
Boy, what a contrast.
So very repetitious, very monotonous, and the sicker and sicker people get, the more the variability collapses, and ultimately you may end up with just one note repeating itself. So in the sickest pathologies, you can literally go from this very musical, tuneful dance, which is the healthy heartbeat, to what becomes a one-note marching band. The essence of healthy function is adaptability, the ability to cope with an environment that's going to play tricks on you. You don't know what's going to happen next. So having this sort of bubbly type of turbulent-like dynamic, which has all these different frequencies, all these different responses built in, gives you an advantage.
So as long as you're playing a symphony in your chest, everything's fine.
It's the old story about a song in your heart, but literally that is in fact the case.
Okay, so that was your Valentine's Day video. Okay. So when we talk about homeostasis, there is a mechanism that our body uses in order to, I can't remember which slide that is, in order to maintain homeostasis. And this is what is referred to as the negative feedback mechanism. And people get confused here because generally negative is a bad thing, right? But in this case, negative is a good thing. Because what this means is that the response of the system to a stimulus is going to reduce the stimulus and bring it back to normal. So negative feedback mechanisms are what maintain homeostasis versus positive feedback mechanisms, which we'll talk about in a second, which do not lead to homeostasis. Okay, so mechanisms. Okay. So in a negative feedback mechanism, we have a stimulus, and it's important to realize that this can be internal or external. So generally, when we think of stimuli, we think of touch or sight or whatever, but it could be like the temperature of your blood. It could be like the calcium levels in your blood. It could be the number of red blood cells that you have in your blood. Okay, so it can be internal as well as external. And then we have a receptor. And a receptor is a type of neuron, it's a type of nerve cell that is able to detect that stimulus and cause a response. Okay, so it is a modified neuron that can detect the stimulus and send a signal to the control center. Sometimes the receptor is in the control center. Okay, I'll give you some examples of those. When we look at the control center, sometimes we think the control center must be the brain, but it is not always the brain. So for example, the kidney is the control center for regulating the number of red blood cells you have in your body. The fat is actually has a mechanism to regulate the amount of fat you have in your body, helps to regulate your metabolism. So the control center is not always the brain, but this is what sends or receives the information from the receptor. And this is where the set point resides. So this is what controls the set point. So the set point could be a range. So it could be the range of your resting heart rate. It could be your body temperature, the range of which we have differences in our body temperature. It could also be the range of your blood glucose levels. So that is the set point. And the control center is what causes a response. By sending a signal to what is called the effector. So we have the receptor, but the effector is what is going to cause a response to decrease the stimulus. So the effector initiates a response that decreases the initial stimulus. So some examples of effectors could be the bone marrow. The bone marrow, when it receives a signal from the kidney, will start to produce more red blood cells. An effector could be the heart, where your heart rate could speed up to increase the supply of oxygen to your muscles. The effectors could be your skeletal muscles that cause you to shiver, or your sweat glands that cause you to sweat. So this is anything that causes a response that is going to decrease the initial stimulus. So in your book, there is an example of thermoregulation. Okay. So in terms of body temperature, we are about 37 degrees Celsius. So Celsius, just like the metric system, is useful because 0 is freezing and 100 is boiling, right? Rather than 98 degrees Fahrenheit, which is what we generally measure in this country as to be our body temperature. So this would be the standard temperature for a human, right? When we decrease the body temperature, this decreases the blood temperature. This sends a signal to the control center, which in this case is the hypothalamus. So this is detected by receptors. Oopsie, detected. In the hypothalamus. So the hypothalamus is part of the brain. It's kind of in the center of the brain. It also regulates hunger and thirst and also fear and sexual responses. Okay. So that sends it to the hypothalamus. So this is my control center. The hypothalamus sends a signal. So this is where the set point is. A signal is sent to the skeletal muscles. The skeletal muscles in this case would be the effectors. Okay. And what do the skeletal muscles do? Shiver, right? So they start to contract. So shivering is the response, right? And this increases body temperature. So when you're cold, you shiver. We have the ability to put on layers of clothes, right? To keep our body temperature. So sometimes before we start shivering, we're usually smart enough to put on something or move to a warmer environment. Okay. So shivering is a mechanism to increase body temperature because as we convert chemical energy into mechanical energy, heat is lost. Okay. So in your book, they have the opposite, which would be when the body temperature goes too high. So when the body temperature goes to high, what happens is blood vessels dilate in the skin and you turn red. And then you start to produce sweat glands. So the sweat glands would also be effectors. And then heat is removed from the surface of the skin by evaporation, and that removes the heat and cools down the body. So in this case, the effectors would be the blood vessels and the sweat glands. So this is what normally happens to regulate our body temperature. And which do you think is more dangerous? Like, which do you think will kill you faster? Becoming hyperthermic or hypothermic? Hyper, right? So if you get too hot, so for example, it's not a good idea to wear black sweatpants, black hood, and then decide you're gonna run in the middle of the day during the hot summer, because you're not gonna be able to evaporate and your body temperature could get too high and you're insulated. And so then that could cause essentially your proteins to start unfold and denature. That is also why when people are, when they drown in cold water, they're much more likely to come back if they are resuscitated within a given period of time than if it is in warm water. Because the cold water actually slows down the metabolic rate, slows everything down, but doesn't actually do thermal damage to the proteins in the body. Okay, so what about fever? So this is not the normal regulation of body temperature. So if I asked you for the normal regulation of body temperature, you wouldn't explain to me fever, right? So fever is a special instance, and it is actually a defense mechanism. So it can actually make it more likely that you are going to get well more quickly than if you actually take fever-reducing drugs, right? So if you have a low-grade fever and can just, live with how that makes you feel, which is pretty shitty, even if it's like 101, right? You feel pretty bad. And you can imagine, that's just a small from 98.6 to 101, you can really tell the difference, right, between those two. But it actually will help to decrease the ability of the microbes to reproduce, and so it helps to increase your healing rate, your getting over the infection. Okay, so what this means when you get a fever is that the set point changes. And this is actually due to chemicals that are released by white blood cells. So white blood cells, or leukocytes, white blood cells release a chemical called a pyrogen. So you can remember this pretty easily because pyro means fire, gen means generator. This is a fire-generating molecule, right? And so your white blood cells say, oh, there's infection, we're going to release pyrogens. Your set point changes, so it increases. So on the onset of a fever, how do you feel? Chilled, right? You feel cold. Right? So you get in bed and you put every blanket you have around you on top of you, and then you have the heating pad, right? And you're cold and you're still shivering. So you tend to shiver. So even though you are hot, right? If somebody touches you, they go, oh my God, you're so hot, you're shivering, right? Because that shivering is, your set point has not been reached yet, okay? So then when this fever breaks, fever breaks, how do you feel? Warm, right? So you feel hot, right? And you sweat. So like, oh, the fever broke, you're sweating. What that means is that the set point has gone back down to normal and your body temperature is too high. And so it senses that your body temperature is too high, so you will begin to sweat to lower your body temperature back down. So you always can tell when the fever, when your set point changes, because you'll feel cold even if you're 100 degrees, and then you'll start to feel hot and you'll have to take everything off and readjust, okay? So there is a limit to, oh, go ahead. Okay.
So what does it mean when you're going hot and cold, hot and cold, hot and cold?
That must mean the set point is changing.
Constantly.
Yeah. And it could be if you take drugs. So Tylenol and ibuprofen reduce fevers. They're fever reducers. Right. And so if you take those drugs, then you're going to reduce your fever for however 4 hours, and then you're going to feel it again. Yeah. So maybe your set point hasn't, or maybe you're just fluctuating around your set point because maybe it's the way you're putting clothes on, taking clothes off. I don't know. Yeah. Question.
So I wonder then, does your body maintain its regular temperature if you're sweating? Like if you're like in a sauna, does the
it tries to
stays the same?
It tries to. So that's why we sweat profusely when you're in a sauna, and that's why you have to be careful. They say take breaks, go back outside, because you don't want to overheat the body. Yes. So yeah. So it tries to maintain the internal temperature, but people can die if they stay in the sauna too long. So you can get dehydrated pretty easily too. And that's why steam rooms are a little bit more easily to get too hot in than dry rooms, because the steam prevents evaporation off the surface of the body. Okay, so the other thing I wanted to mention is the limit. So there seems to be a limit, and it's about 104 degrees Fahrenheit. Okay. After that, you start to get problems. So you have to watch your fever, and if it gets over 104, which generally is in children, because children aren't able to adjust, babies and young children aren't able to say when they're, take their clothes off, put their clothes on, they might be stuck in whatever you put them in, right? And so after 104 degrees, this can lead to seizures. Okay, so the scariest thing is when you have a young child that starts a seizure and you're freaking out, and then you call the doctor and they say, oh, it's okay. What's their temperature? Oh, that's okay. Put them in a tepid bath, right? So you take their clothes off and you put them in a cool bath, and that will cool them back down. So it's pretty common for young children to have seizures when they get too hot, okay? And then they also say give them ibuprofen or Tylenol, and you can give them both, right, to keep the fever below 104 degrees. Okay. Has anybody ever experienced that in themselves, or just your child?
My daughter hit 6 months. So we're pretty freaked.
Yes. And did you take her to the hospital? And yeah, yeah, yep. That's pretty high.
You didn't see it. We were freaking out.
Yeah, yeah. Okay, I guess with this flu season, I think children this flu causes really high fevers for some reason. So you gotta be careful. Okay, so that is a defense mechanism. This is not what normally is observed in the body. Well, it is normal, but it's, because of an infection. Okay, so when we look at a positive feedback, this does not maintain homeostasis. Okay, does not. Because what this means is that the response is going to actually increase the stimulus. So the homeostasis. So the response actually has a positive effect on the stimulus. And so there are just a couple of examples. One of this is labor, right? Giving birth. The other one is blood clotting. Blood clotting is a little bit more complicated to explain than labor. So they use that in your book. So notice that in labor, there is an endpoint when the baby comes out, right? So you're not trying to maintain a constant internal environment. You're trying to push that baby out. So the baby, when it pushes on the cervix, that pressure on the cervix actually causes more oxytocin to be released, which actually causes stronger contractions. So the contractions start out really minor, and then they build, and they build, and they build, and they build until the cervix opens up and the baby is able to pass through the birth canal. Okay, so this is the response increases the stimulus. And in blood clotting, there is an end. We don't want the blood to stay clotted, but it is kind of a positive feedback where that once it starts, it tends to build on itself. So blood clotting is another example. Okay, so positive does not mean good in this case. The negative is the one that controls, that controls homeostasis. Okay, so we're going to be talking about the different systems, and I'm just going to introduce the topic and talk a little bit about the other phyla, and then we're going to focus on humans. Okay. So when we look at digestion, there are two things that we need to get from our food. We need to get nutrients, which are building blocks, and we also need to get energy, which is measured in calories. So you can have a diet that is high in calories and low in nutrients, right? So if you drink a lot of alcohol and eat a lot of potato chips, for example, so like beer and potato chips are like your main source of food, then you would have a lot of calories, but you would actually be malnourished because you don't have the appropriate nutrients, okay? So those are two things to take into consideration when you think about diet and what digestion is all about. So we as animals obviously have to take in the nutrients. We're not capable of building energy. And then we can use some forms of food as energy. And so we'll talk about the different types of food in a minute. We also have what is called intracellular digestion. So if you look at this word, intra means inside, okay? So this is versus extracellular digestion. So we have, as animals, have evolved to have a space inside our body where we secrete enzymes and we digest the food outside of the cell. If you think about sponges, for example, sponges do not have a digestive tract at all. So they only have intracellular digestion. When you think about jellyfish, they're the first ones with a mouth and an anus, it's one opening. And they are able to eat larger items of food because they break it down first extracellularly. So that's outside the cell and in a cavity. And so if you think about this, this is quite amazing because I have a stomach that is full of acid. That acid would do damage to my body if it got loose. I also have my digestive tract is full of enzymes that could eat my cell from the inside out, right? So that extracellular digestion is quite amazing that it takes place inside my body, and I could, digest my own tissues, but I don't, right? I'm digesting the food that I take in. So if we think about organisms like the hydra and the jellyfish, they have an incomplete digestive tract. And then when we look at this would be like a nematode, a roundworm, we see that they have a complete digestive tract. And the advantage of the complete digestive tract with two openings is that we have compartmentalization. So we have different things, different types of digestion, different things happening in different parts. So now we can have a specialized digestive system, okay, with complete digestive tract. Okay, so let's look at the human digestive system. So we have, obviously, a mouth or an oral cavity. That's an oral, not coral, oral cavity. Okay. So one of the interesting things about our oral cavity compared to like fish or compared to like frogs is that we actually have our nasal cavity separate from our mouth. So this is actually separated from our nasal cavity. If you are looking at a frog, they do not have a separate nasal and oral cavity. So they have to breathe and eat the same time. They're not separated. So our oral cavity is separated from our nose, which means that we can chew our food and breathe at the same time. Okay. So we have a heterodox dentition. What does that mean? Right, diversity in teeth. This is allows for more efficient mechanical digestion. Okay. If you ever see things coming out in your feces whole, like peanuts or corn, that means you did not chew your food properly, right? So things can sometimes make it through without getting digested if you don't chew it. Okay, so that's heterodox dentition. We also have a tongue. And the tongue is useful because it can manipulate the food. And it creates what is called a food bolus. So this is the old saying that you do not want to inhale your food, right? You don't want to like breathe in your food because it could potentially get into your respiratory tract and kill you. So you want to manipulate your food, you want to chew it, you want to calmly put it into a bolus, right? And then you want to swallow it. And that food bolus is really important because it pushes down on the epiglottis. There's a mechanical valve and the food bolus pushes down on it and it makes the food go to the right tube. It makes it go into the esophagus and not into the trachea. So I'll show you a diagram of that in a second. So the food bolus is really important, that you swallow your food in balls, right? Big chunks of it. Okay, the tongue is also taste, right? And this may not play much of a role today because we have, you know, the FDA, we have nutritionists creating, you know, food for us that says it's supposed to be healthy, so I eat it, right? But if you think about in the past when we had to forage, taste was probably really important, right? So that you could taste and you could crave certain nutrients. And so we generally have what is salty, considered salty. So that would be minerals like sodium and chloride. So I need sodium in my diet to a limited extent. We have over-salted most of our processed foods, however, right? So we get too much salt. So this is sodium and chloride, which are important electrolytes. Those are ions, so that's what makes up salt, sodium and chloride. We also can taste sour. What do you think, what vitamin is associated with sourness?
Vitamin C.
Vitamin C, right? Have you ever really craved vitamin C, right? Like, oh, I need some vitamin C, right? And you're just like craving it, right? Could be that you need it. This is a nutrient that we have to have in our diet in order for collagen to be formed in our body, which is important in the skin, it's important in our bones, can lead to scurvy if we do not have vitamin C. Okay. We also have bitter. And bitter is an interesting one because in general, bitter means toxic, right? Because plants produce bitter chemicals to try to prevent herbivory. So most plants that or produce a chemical so that if you taste it, it could be a toxin and you would spit it out. But caffeine is an example, dark chocolate, we actually start to like the bitter taste because we associate it with influences on our nervous system. So toxins also affect the nervous system, right? So we can crave coffee because it speeds up our nervous system, right? It gives us that caffeine high, even though it's bitter. Sour is actually a measure of pH, which is important because pH can affect the way the enzymes work in your mouth. So what happens when you even think about eating a pickle? You salivate, right? And that saliva would actually decrease the pH. So it actually neutralizes the pH so that you can still digest that food in your mouth, right? So this is, salivation is just a way to neutralize the pH in your mouth.
Is it bitter? Like, doesn't it have some effect on pH as well?
I don't really think so.
Like more alkaline things taste bitter.
Like a base, like sodium hydroxide. Yeah, I don't know. But that, yeah, so sour is more acidic. Yeah, I don't know have to think about that one. I guess, so baking soda water tastes bitter, kind of. Yeah. Okay, and then there is another one that just was recognized. And does anybody, oh, sweet, sorry. Okay, if I'm running low on calories and energy, so this is why, like, if you don't eat all day, you want something sweet, right? Because you're like low on energy, so then you're going to eat something sweet. So this is why they say that if you eat regularly, then you may not crave sweet as much. Okay, and then there's one more. Does anybody know what it is?
Umami.
Umami. Okay. Umami is savory, and it is associated with protein, right? So like you want a savory steak, right? The problem with this one is it has actually been hijacked by the junk food industry to make you think that you're getting nutrients when you're really not, right? So what is that chemical that they add to a lot of food? Oops. Does anybody know?
Sodium glutamate.
Yes, MSG. Monosodium glutamate, MSG. And there's big controversy over whether MSG is bad for you or not, but it can trick your body. So for example, Doritos, right? You're craving something savory, you need some nutrients, you're eating Doritos, your body thinks, your mind thinks you're getting protein. There's no protein in Doritos, right? So you'll just kind of keep eating it and eating it and eating it, and you'll never get the protein, so you'll just keep eating it, right? And so it just kind of hijacks the body's ability to recognize what is protein and what is not, right? So taste is a really important component because it gives us information about the nutrients of the food, right? What is in it? Okay. We also have salivary glands. Salivary glands release water, right? Water is necessary for chemical reactions to occur in your mouth. Chemical reactions are occurring in your mouth. We have enzymes. And one of these is an enzyme called salivary amylase. Anytime you see this ACE ending, that means that you're looking at an enzyme. Not all enzymes, however, have an ACE ending. So like pepsin is an enzyme in your stomach. It doesn't have the ACE ending, but if it has the ACE ending, it is an enzyme, okay? So salivary amylase is an enzyme that is in your saliva, and we're gonna look at that in a second. We also have antibodies, and what do you think the antibodies function as? Immune, right? Immunity. So that would be like immunity against bacteria, fungi, stuff that we have been exposed to previously. So we take in a lot of microbes when we eat. Right? It's all over the food, all over our hands. Yep.
So do those work like against food allergies?
You do not want to produce antibodies to nutrients. So like antibodies to gluten or antibodies to peanuts, you don't want those because that's an allergen, and then your body is acting inappropriately towards them. So these antibodies, I don't think are the ones that are associated with allergies. They're IgA antibodies. It's the IgE antibodies like gluten is a problem, yeah.
And do these function against toxins too?
Yes.
Like,
Yes.
because you said immunity, but like you were starting to.
Yeah, I'm trying to think of an example of where you would you'd have a known toxin. Maybe bacterial toxins. They would act against those too. Okay.
So,
Yep.
What could cause the production, if you didn't produce or you weren't allergic to something before, what could cause the sudden production of those antibodies against, like, say, gluten?
Gluten. That's a kind of, we don't quite know the mechanism of that. That one hypothesis that I gave you is that your immune system doesn't have anything to fight off. So it learns to fight off stuff like the hygiene hypothesis, or, the hookworms actually decrease your reaction to gluten. If you had a hookworm, you might not be allergic to peanuts anymore, right? Because your immune system is fighting off other things. So it's the same antibodies that fight off worms that cause the allergic responses to food. Yep. Yep. Okay, so we have the salivary glands. So I'm going to give you a little handout, and I want you to read through it. And so you'll read through this first part, and then I want you to answer the questions on the back. And so this is just group work to do as a little break from lecture. So you can read through it together or alone, separately, and then get together with another person in class and answer the questions. And if you need to in the bathroom, or get a drink of water. I'm also going to give you a handout of the digestive system. Okay, so what does salivary amylase break down?
Starch.
Starch. So it breaks down starch into a molecule called
Maltose.
maltose. And maltose is actually a disaccharide. So it's a simpler sugar, so you can actually taste maltose as sweet. So if you've ever eaten bread when you were maybe even as adults, or as a kid, you know, you hold it in your mouth, and the longer you hold it in your mouth, the sweeter it gets. And the reason for that is that starch itself is not sweet. So if you think about eating a piece of raw potato, it does not taste sweet. But if you held that piece of raw potato in your mouth long enough, your body would start to break that down into maltose, and it would taste sweet. Okay? And so then where does that maltose go? Into your small intestine. And what other enzymes work on that? Maltase, right? So that's easy to remember. So maltase breaks down maltose into what? Glucose. So glucose is the primary form of energy that we use in our bodies. Okay? What other enzyme do we have there? There's one more.
Pancreatic.
Pancreatic. So there's actually two glands that produce amylase, the salivary glands and then the pancreas. So the pancreas is a gland that sits right by your stomach, and there's a duct that goes and transports enzymes to your small intestine. So that is any starch that doesn't get broken down. Let's say you eat it really fast and it doesn't stay in your mouth very long. By the time then it gets down to your small intestine, you can also break down the starch in your small intestine. And then glucose is what is taken up by the individual cells. So we have to have the maltase break it down into glucose before the glucose can be taken up intracellularly. Okay? So then we found out that there was a particular gene that codes for this amylase, and it's called AMY1, right? And we have a diploid gene copy number. And so what was the range? 2 to 15. And so we can see here that when we look at the number of individuals in each population, so let's look at the European population. So this is European. And so you notice that if we look at like eight copies, so this is the proportion of individuals. So about 80% of the population has 8 or more copies, okay? So what that suggests is that we relied at some point in our past on a diet that was high in starch. So if you compared this to people that were primarily meat eaters that did not rely on starch, then you would predict that they would have a lower number of amylase genes. So we became, you know, farmers, and we relied upon potatoes for our nutrients so that we would be better able to digest it. But this all kind of relies on the fact that you would have to first see whether or not an increased number of the copies of the genes actually translates to an increased amount of salivary amylase. So how would you go about number 4? What would be an independent variable? Yes, the number of copies would be your independent variable. So you first test them genetically, right? And then you would look at how much salivary amylase they had. And so that would be your dependent variable. Okay. Control variables might include such things as you would want to look at diet, population. You might, oh, I didn't realize you were here, I'm sorry.
Oh, it's fine.
Diet, population, right? Maybe even age. Age might affect salivary amylase production.
Those are control actually.
Yes.
Would you say the dependent was?
The dependent would be the amount of the enzyme. The independent would be the gene. So we would say that the amount of enzyme is dependent upon the number of the copies of the gene that they have.
Independent copy then?
Yes, independent is the number of copies. So if you can digest starch better, like if I didn't have any amylase in my body, I would not be able to digest starch at all, right? It would just go through my whole body and I would never be able to get the glucose out of it.
Did that make you sick though?
You would probably die because you have to,
Say like something would happen.
I mean, you wouldn't maybe die, but you would have to eat simple sugars. You could not rely upon complex sugars for your glucose. And you can also convert fat to glucose and other things to glucose. Okay. Any questions about that idea? How our digestive system and our ability to digest is dependent upon our past evolution, what we depended upon for food. Okay, did everybody get a copy of the handout? Of this handout, yep. Okay. So these are all salivary glands. There's one that is in your cheek, which is the largest one. You don't need to know that. That's called the parotid gland. And when you have mumps, that is what it gets infected. So that's why when that virus infects the parotid gland, the salivary gland, it causes you to get like chipmunk-like pouches on your face. We all probably know that there's one underneath our tongue. So that's the sublingual one. And then there's one underneath the mandible that you can sometimes feel. There's also lymph nodes underneath here, but that is also the salivary gland. Okay. This is the hard and soft palate. That separates the oral cavity from the nasal cavity, which means that we can chew and breathe at the same time. And then obviously you see the tongue in there. They don't show the teeth. Okay, so what would this structure be right here? Esophagus. So you can't feel the esophagus, but if you feel the front of your throat, what are you feeling? Your trachea. So the trachea actually sits anterior in front of the esophagus. So when we do the dissection of the rat, you're going to need to pull away the trachea to see the esophagus behind it. So it is posterior.
Is that what we're doing with labs?
I think so. That's what we're going to probably do. One structure that is not shown here is the structure right here, which sits like a dome above the liver. And what is that structure? The diaphragm. Okay. The back of the throat, before it branches into the esophagus and the trachea, so the back of the throat, right, like right here, has another name. Does anybody know what the back of your throat is called? It is called the pharynx. So the pharynx is actually the common opening to the digestive and respiratory systems. So if we think about the primitive chordates, they had pharyngeal gill slits. The pharyngeal gills captured food particles and also were used in respiration. So the reason why we have this common opening, which is probably not the best system to have, because people all the time choke and can die from choking when they get up into their respiratory system. That's probably not the best way. It's probably better to have separate openings, but that just is the way that we are designed. Sometimes they'll do a tracheotomy where they'll actually cut a hole in the trachea so that you bypass the pharynx so you can breathe right through that opening into the respiratory tract, right?
You see that in people that like smoke cigarettes a lot and then they like get cancer, so it affects their whole.
Right, yeah. They get throat cancer, yeah. Sometimes they also get esophageal cancer and have to have the parts of their esophagus removed too. So the esophagus is an interesting structure because it has peristaltic movement. So we've seen peristaltic before when we were talking about the movement of the earthworm and its ability to burrow using its hydrostatic skeleton. But this is the alternating contraction and relaxation of the esophagus. So what type of tissue probably allows for the peristaltic movement and the pushing the food down your esophagus towards your stomach? Smooth muscle. Is smooth muscle voluntary or involuntary? Involuntary, right? So there's longitudinal and circular, just like muscles, just like with the earthworm, there's longitudinal and circular muscles around our esophagus, and it contracts and relaxes and it pushes the food down. And you probably have experienced like swallowing a chip before you properly chewed it, and you can feel it scratching, right, as it comes down. And then you might have also experienced swallowing a pill and having it stick in your esophagus, and it kind of drives you crazy because what you're sensing there is that the esophagus is trying to move the pill down, and the pill is stuck to the side, right, and it cannot move. And so that's why it feels annoying to you. That's that peristaltic movement. Okay, so we have here where the esophagus penetrates the diaphragm, we have what is called the Gastro Esophageal Sphincter. Because we want food generally to move unidirectionally through our digestive tract, we do not want to have to regurgitate unless we're vomiting up and it's a toxin that our body has sensed, right? So once we swallow, we don't want stuff coming back up the esophagus. So this is a sphincter that aids in that. And it is actually where kind of one function of the diaphragm is it actually helps to create that sphincter. Okay.
Does that gastroesophageal sphincter work when you get people that have acid reflux?
Right, and so the problem is when you have acid reflux. Right, so you get acid into the esophagus. The esophagus, unlike the stomach, is not protected from acid digestion. And so the big, people say, oh, I have acid reflux, but the big problem with acid reflux is that if you have it your entire life, the older they get, the more freaked out people get by it, the doctors, right? So I had acid reflux, and my doctor told me never to go off my acid reducers. And why is that?
Because the longer that you're off of your acid reducers, your acid is eating away your esophagus.
And what is going to happen to my esophagus?
It's going to deteriorate.
And it's going to get cancer.
Yeah.
Right. So acid reflux can lead to esophageal cancer, right? So you want to protect yourself from that, right? So how do they detect esophageal cancer? Has anybody ever had that test done? Yeah, so they do an endoscope. So they put a little scope down through your mouth and into your esophagus, and they go down, and what they're doing is they're looking for precancerous cells at the margin where your esophagus enters into your stomach. Yeah. Now there is a problem sometimes with people having a defective sphincter here, and this is what is called a hiatal hernia. So when we think of a hernia, we think of like something protruding out the body wall. Like we think of like an abdominal hernia where you get damage to the abdominal wall, part of your intestine is poking out, right? This one, a hernia just means that it is protruding out, right? And hiatal is, I think it's where the esophagus and the stomach meet. And so this is where the stomach is above the diaphragm. So just part of stomach. Part of stomach is above the diaphragm. This can sometimes happen with females when they're pregnant because the baby takes up so much space that it pushes the stomach up, right? It makes it more difficult to breathe. They can also lead to the hiatal hernia, right? And then sometimes it just seems to be people get predisposed to it. Maybe it runs in a family. So if we look at a diagram that shows that hiatal hernia, oops, there it is right here. So this is a diagram that shows the diaphragm. This is your esophagus, and this is your stomach. So notice just that a part of the stomach that is producing the acid extends above the diaphragm so that the acid can then easily get into the esophagus. And so that's one thing that they might give you acid reducers for. The other way that they treat this, which unfortunately doesn't tend to last, is they go in and do surgery and they pull the stomach down and they try to kind of wrap muscles around there to keep the stomach from extending up in the diaphragm or up above the diaphragm. So you can have surgery to repair a hiatal hernia. Okay. So the next organ that we're going to look at is the stomach. And the stomach has a function in that it stores food. And so it can store up to like 2 liters of fluid and food, about 2 liters. So if you did not have a stomach, you would not be able to eat a large meal, right? You would only be able to eat little bits of food all day long. So when people have their stomach stapled, or when they have a gastric bypass, or when they reduce the storage capacity using like a belt around the stomach, they're just preventing themselves from eating large meals. But it doesn't mean that you can't eat a lot of calories over an extended period of time if you continually eat, okay? So it stores food, and that's one of its main functions, yeah.
Is there any truth to the stomach shrinking if you cut back? Like, does it?
Yes, so it kind of shrinks up, yes. So it just becomes smaller, and it's stored. So if you don't eat large foods, then its capacity shrinks, yes. Yeah, there is some truth to that. Okay, the other function of the stomach is mechanical digestion. Because the stomach is also smooth muscle. And so it churns the food and mixes it with gastric juice. When this actually kind of starts by just seeing food. So I can see food, I can smell it, and then my stomach will start to churn, right? And that's because it's getting ready, right? It's also going to start to produce enzymes. And so if I don't eat, I'm going to have all those enzymes in my stomach, which kind of have nothing to do with maybe some acid too. So that growling of your stomach is just, it's starting to churn in preparation for mechanical digestion, and it growls because there's no food in there. Right. Yeah.
So what happens when you don't eat and then like your stomach just continues starts to growl and like your body starts to eat away from fat cells? Like how does that entire
we'll talk about metabolism and digestion and hunger, but at the very end of the section. So probably not today. Okay, so it churns the food. When we mix gastric food with chyme, so gastric juice and food, when we mix it together, this is called chyme. So food plus gastric juice equals chyme. We also have chemical digestion in our stomach. So the chemical digestion breaks bonds between molecules. And so one way that we break bonds between molecules is by using hydrochloric acid, HCl. This is actually a very strong acid. So it can be like a pH 2 in your stomach. So this can actually break down and destroy microorganisms. So it can kill microbes and fungi. It can actually eat iron, right? Nails, it can break it down, right? So you can digest that, okay? You actually accidentally ingest it, okay? The hydrochloric acid is especially important with meat digestion. Okay, so because I take an acid reducer and I don't eat a lot of meat, when I do eat meat, I have a really hard time digesting it. It feels like it stays in my stomach for a long period of time. Because what the hydrochloric acid does is it breaks down the connective tissue that holds the muscle. So it breaks down connective tissue and it denatures protein. So denaturation of protein just means that it unfolds the protein in preparation for enzymes getting to work. So this is denatures means to unfold. Right, so eating steak can be very painful for me to do. Yep.
So whatever like the digestion can get all that stuff. So how do we get poison? So we eat something bad and does it even live or something?
Sometimes it can get digested, like if it's E. coli, so if it's a bacteria, sometimes the bacteria can get into the bloodstream and then it makes you sick. It actually, your medulla oblongata in your brain can detect toxins and microbes in your body, and that is what causes you to vomit, right? So it's detected that there's been an infection and invasion. Yeah, that's a good question. So not all microbes, and we'll talk about that in a second, because ulcers now are thought to be due to microbial infection. So not all microbes are a problem with hydrochloric acid. But I have to tell you that I got really sick in Costa Rica because because I take these acid reducers, right? And my theory is that I was less protected from the bacteria in the food than my children or my husband. So my husband and children did not get sick at all. But I was like, every time I eat someplace, I would vomit, right? It was like, it's awful. But anyway, so maybe it has to do with the levels of hydrochloric acid in your stomach. Yeah. Okay, so that's chemical digestion. We also have a enzyme called pepsinogen. This is in its inactive state. So this is inactive. It is converted to pepsin in the presence of hydrochloric acid. So hydrochloric acid converts pepsinogen to pepsin, and this is active. Pepsin is an enzyme, but it does not have the ACE ending, but it breaks down proteins by breaking the peptide bonds between amino acids. So hence why it is called pepsin, right? And all of a sudden, I just saw that Pepsi in there.
Yeah, I was just
They say if you have stomach aches, you drink Pepsi and it helps your stomach, but I'm not sure why.
Also, I don't know if you've ever seen the videos where they take a chunk of meat and they'll put it in Pepsi or like Coke, and it will go back and it'll be gone.
Digested because it's acidic.
Or it turns eggs to.
Yeah, so it is an acid, but that's because of the carbon dioxide that's because of the fizz. But I remember as a child that there used to be something about Pepsi, that they used to sell syrup. I don't know. It just hit me. I don't know where that Pepsi, where that word came from. Maybe yes, Pepto-Bismol. Pepto-Bismol is good for coating the stomach and reducing acid indigestion. So this breaks the peptide bonds between amino acids. I'll have to look that up about Pepsi. Breaks peptide bonds between amino acids. Okay, so if pepsin digests protein, well, the stomach has protein in it, right? This lining of the stomach has protein in it. So this is probably why it first comes out as an inactive form, and then it becomes active. So this helps to prevent self-digestion. So that process prevents self-digestion, like the digestion of your digestive tract, right? There's another mechanism, which is the mucous membrane. Does anybody remember what type of cells in the lining of the epithelial tissue actually produce mucus? Goblet cells, right? And the acid cannot eat through the mucous membrane. So as long as you have a healthy mucous membrane in your stomach, you will not tend to get ulcers. So when we talk about gastric ulcers, right, this is when you get sores in the stomach. And they can bleed, and they could be actually a source of anemia. So if you have too many stomach ulcers, you're bleeding out in your stomach, right? You could vomit the blood out also, but you're bleeding, and that could make you anemic because you keep having to produce more blood. So the sores in the stomach, in the past, they said, oh, it is due to stress, or it is due to diet. The diet probably does play some role in this gastric ulcers, because some things can break down that mucous membrane and be absorbed directly across the surface of the stomach. So for example, alcohol. Okay, so the worst thing you can do for your mucous membrane is to go home after all day not eating, or haven't eaten anything, and you eat potato chips, drink a beer, and take a couple of aspirin, right? Worst thing ever. Total worst thing. So that's why they say don't drink alcohol on an empty stomach, right? If you eat first and then drink the alcohol, it's not going to do as much damage to the lining, right?
And I mean, like, eat something that doesn't have
yeah, it's not potato chips or salty, right?
What does the aspirin have to do with it?
Aspirin just does make your blood, that's why they said don't use aspirin for the longest time. Now aspirin is kind of coming back because it's supposedly good for your cardiovascular system. When I was a kid, we didn't even have ibuprofen or Tylenol. That's all we had is aspirin at home. Okay, but there is also a bacterial infection that could be the problem. And I said that most bacteria are killed by the acid, but there is a strain of bacteria called H. pylori. It's actually Helicobacter pylori. So that's just the genus and the species name of a bacteria. This is resistant to acid, so it lives in your stomach. So now, if you ever have gastric ulcers, for an extended period of time, and it doesn't matter what you eat or how unstressed you become, they will do a test to see if you have H. pylori. And you don't really want it because these are really resistant to antibiotics, and so you end up taking antibiotics for like 6 months to get rid of this infection. It takes a lot of time to get rid of it. I think that they can even do a breath test. Used to be they can test to see whether or not you have H. pylori by doing a breath test. Yeah.
Do gastric ulcers, like my sister has, when she can't eat spicy foods at all? Like, do you know how that?
Spicy foods typically are not a problem because the spice in the food actually is not actually doing any damage to the tissues. It's just making your body think it is. But it might be that there's acid in there too. So it could be that. So maybe if she's eating salsa, it's tomato-based salsa, and it's probably the acid more than the spice. Because actually, for spiciness, like if you have throat cancer, if you have mouth cancer, they actually give you capsaicin. They give you the spice in like a lozenge, and it actually capsaicin mimics tissue damage and pain, so you actually produce endorphins, so it actually relieves the pain of it. So the pain part of it could actually be relieved by the capsaicin. Okay, so I think I have a picture of, yes, and so here's just a diagram showing the layer of the mucus and then the bacteria that are able to survive. Some people supposedly are carriers of H. pylori and never have a problem with it. So that's another weird thing, is that some people have it, but it doesn't cause the ulcers. Some people have it and it does cause ulcers.
So you think it might be something that causes them to, like,
Yeah.
trigger.
Yeah, and they also think that if they get into the blood, they also could lead to cardiovascular disease, like blockage of blood vessels too. So they want to keep it out of your circulatory system too. Okay, so I'm just going to stop there for today, and we'll start on the small intestine, and we'll finish up digestion on next Monday. So I will see you tomorrow for lab.
Yeah,